you know, even in healthy developing brain, there's a balance between your genetic makeup and your environment. And one of the places that those interact almost in a very 50-50 manner is actually in that microbiota population that lives in your gut. And so I'm going to tell you a little bit from the perspective of how does your gut bacteria influence behavior and brain structure, but I could tell you a lot more. I'm going to try to keep it as short as possible so we can answer the questions that you have, but it really is using your gut bacteria or signaling pathways between your gut and your brain might actually be good biological readouts for some of the questions that we currently can't answer from a top-down perspective. So my lab's very interested in how the body influences the brain, and I'm going to tell you a bit about that. So I'm going to give you a little bit of introduction, and I'm going to show you a bit of research data, and I'm going to do it in as brief a story as I can from a detailed perspective. So microbiota, if you haven't heard about them, are the gut bacteria that are in your gastrointestinal tract. They're in your lungs, they're covering your skin, they're in your mouth. They're there because they're friends of ours. They're not there as pathogens. They're different from the ones that cause viral disease or cause pneumonia. And when we think about them, they've been in the news a lot in the last 5 years, probably more than ever before. But they're equal to us in number. So we have as many bacterial cells in our body as we have body and brain cells. So they're a one-to-one partner in life. So, you know, it used to be we thought there was 10 of them to every one of us. Now we think it's one-to-one with some new math tools. But what's really clear is when it comes to the genetic load, that is the DNA that informs how the biology of your system works, there is thousands times more DNA in the bacteria in your gut than there is in your whole body. So that's important from a clinical perspective because if we look around the room, we're all about 99% similar with respect to how we're compared at the DNA. So if we sequenced everybody's genome in the room, we would have an overlap about 99%. So some of these places where genetic differences are driving disorders are in that 1%, and maybe it's a little bit bigger in some populations. But the bacteria that we share, so how similar is my bacteria to someone else's in the room, is about 10%. So this really is a target for understanding what underlies individual differences. And one of the big themes of POND is how can we use biology to separate out children across disorders, across age, to get more homogeneous groups that we could target a particular therapy to and improve outcomes. So that's the clinical driving force. So what do we know about microbiota? So we know that there's 100 trillion viable bacteria in our gut. And I'm speaking of microbiota with respect to the gut bacteria. They're everywhere else as well, but most of the work goes on in the gut, and most of them are in the gut. There's thousands of species represented. That doesn't mean we each have 1,000 species, but we could. And I think it's up to 4,000 now when we look at it. There is very high interpersonal variability. That means my bacteria are different than yours. But if I looked at my own bacteria today and again in a year from now and again in 2 years from now, it would look pretty much identical. What would be different is if I looked at it at 1 year of age or after a dose of antibiotics or as I age. So it's sort of an interesting thing how we have no bacteria when we're born. We gather it from our mothers as we go through the vaginal tract, and then we get this diversity that grows in the first year of life, and then it changes continuously, just like our brain changes till we're young adults, then it stabilizes, and then as we age it sort of loses its diversity and its function. And my lab would tell you that's linked to the immune system. I'm going to talk a little bit more about that in a minute. But it's essential, first of all, to host pathogen defense. So this is good bacteria that fights off the bad bacteria. It's the reason we can eat some of the foods we choose to eat, like rare meat that's full of bacteria, because these good bacteria are there that say, sorry, this is my home. They're very territorial. It's also very important for nutrient uptake. So if there's any vegetarians in the crowd, you can thank your gut bacteria because you wouldn't be able to metabolize those plant polysaccharides without your gut bacteria and the enzymes and the products that they produce. And like, I'm the only one, actually there's about 10 of us in the world that will put our central to brain development on the introductory slide, but that's the work that my lab's shown and other people that I know. But most importantly, an individual's profile is related to genetics, age, sex, and diet. So even what we eat as babies or the type of food that we eat as young adults or at adolescents in particular can shape what is that composition. So while it stays stable over time, if you change your diet, you can change the profile of your gut. If you change your behavior, you can change the profile of your gut. And this is just for particular, we are colonized at birth. There might be some bacteria around in the placenta, but it's not relevant to brain development, I don't think. And it continues through development. But interestingly, that first year of life where that colonization happens looks very different in a child that was vaginally born versus a C-section baby, and also looks very different whether you're bottle-fed or you're breastfed. So this isn't a problem to think about. It's not a causative factor, but it just tells us that these factors really do influence the development of the microbiota, and I will tell you that that influences the development of the brain. So one of the risk factors for C-section babies is asthma. So one of the things that people try to introduce in some of the pediatric populations is to put the probiotic on board in order to reduce the risk of asthma if the mother has allergies on board or if there's C-sections. And there's a lot of work in pediatric non-neural systems looking at how probiotics can be a benefit. So more recently in the area of brain health, there's been quite a few developments, and in particular we have studies that have examined the effect of these probiotics. So probiotics are live bacteria that if you take enough of them, they have a positive health benefit. And so you can find them in your grocery store. A lot of people take them. But there's been actually systematic studies that show that they can have a benefit to your stress stagings even in healthy individuals. And much of the work has happened in healthy individuals because we really have to know what the normative control is before we can figure out if it's going to fix something that's broken. And so almost every study that has looked at probiotic benefit in a health condition has an impact on anxiety. So what I study in my lab is how the immune system in the gut influence anxiety, but the beneficial expects to be in the anxiety domain, which is a cross-disorder domain for neurodevelopment, but it's also a cross-disorder comorbid condition across the lifespan. Quite often in a lot of medical disorders, anxiety presents first. So this whole body-brain connection might be part of reducing risk. And so there's several studies that have showed a link in healthy individuals to improve mood, to brain activity and emotional centers, and to see benefits of reduced stress proteins in people. These are all in healthy people. And this is some news that came out of Emmer and Mayer's lab at UCLA a few years ago. And what he showed is he took a group of healthy women and gave them a commercially available probiotic drink for 4 weeks, scanned them in the imaging in the MRI before and after it, and showed that their overall resting stress circuit had a dampened effect. So this is healthy individuals. And one of the problems with translating the healthy individual treatment into a population that has a mental illness problem is the fact that the mental illness brain might not look like the healthy brain. So we have to then consider what's the timing and the appropriate use of these in individuals with mental health issues. So I'm going to switch back now to the mouse, because that's actually my expertise, and tell you a little bit about what we do in my lab. So I'll start by saying there's a lot of different ways that we can manipulate the microbiome. And what that means is all of these sort of mirror something about regular life, right? So if I change the diet of a mouse, if I give a high-fat diet, it has a different profile than if I give a low-fat diet. If I give a beef diet, it really enriches some of these bacteria that you might find in the probiotics on the shelf in the grocery store. If I change the genetic strain, or if I modify genetics based on things we know might be implicated in neurodevelopmental disorders, I can change the profile of the bacteria in the gut. If I stress a mouse, it completely changes the profile of the gut. I'm going to show you some of the housing work. If I give them a probiotic, I can change their behavior, whether they're a healthy mouse or if they're a sick mouse. And antibiotics completely change the structure of the gut. And of course, when you give an antibiotic, if it's a short trial, once you take that antibiotic, everything goes back to normal and goes back to that pre-existing state that it was. So we know a lot about all of these different components. But the starting point I'm going to tell you about is one of the ways in biology that we can find out why is something important is by taking it away. And so about 50 years ago, they developed what's called the germ-free mouse. And so this is like the mouse in the bubble. So this is an example up at the top right there of a bubble at McMaster University where we house our germ-free mice. And so these mice have been initially derived by C-section into a sterile mouse, and then they're bred in this sterile environment, so they never see a bacteria of any case. And one of the things that's interesting about these mouse, they're a little bit, they're skinny, they're a little bit hyperactive at times, is that they're missing the adaptive immune system. So they don't have those leukocytes that make your antibodies. They don't have those leukocytes that prevent against pathogens. But they also are resistant to high-fat obesity and a whole host of other things. So they help us point the arrow in the direction of what the microbiome might be influencing with respect to behavior and function. And so this first paper came out about the time I started at Mac by a Japanese group, and it looked at this stress axis. So on the left-hand side here, I show this pathway. So if you have a stress, whether it be an infection, whether it be psychological stress, whether it be sort of an imbalance of the way you cope, there's signals that go to your brain, and your paraventricular nucleus of your hypothalamus produces corticotrophic releasing hormone, which is then signals the pituitary to produce adrenocorticotropic hormone, which goes down to your adrenal gland and releases cortisol in people and corticosterone in mice. So this pathway is very well documented. And what these investigators did is they looked at this pathway in mice that were germ-free following a stressor. So the hatched little sign there is the 1 hour of stress, and then they sampled the blood over time following that stressor. And what you normally see is if you stress the mouse, in this case it was a restraint stress, so you just put them in a tube or some sort of device to allow them not to move around, they don't like it, and if they don't like it, they increase their cortisol or their corticosterone the same as we do. And so after 30 minutes, there's usually a peak of both of these hormones, and then in the germ-free mouse, it was exaggerated. So that's suggesting in some way that your microbiota might be influencing your stress system. So in my lab, what we did is we actually, I forgot to put this in there. So an important point of their studies was they also then took the germ-free mouse and fed it only one bacteria, which is this bifidobacterium, over adolescence and into young adulthood, and then tested the stress system. And when they did that, I'll just point your attention to this one column, is by normalizing or by feeding the germ-free mouse just this one good bacteria that helps, normalize its immune system in other parts of its body, they actually prevented this exaggerated stress response. And in a lot of animal work, they've done this. So if you actually look at, models of liver disorder, models of asthma, other stress models where we stress mice early in life and then test them later in adulthood, if intervening between the stressor and the behavioral test, you feed a probiotic, you often alleviate either the depressive-like or the anxiety-like symptoms or this stress reactivity circuit. So it's not just behavior, it's also got a brain signature. So my lab decided to do some behavior work in these mice, and up in this top corner here, there it is a mouse, and this is an elevated plus maze. So this is just to remind you that in mice we have these very easy tests that are approach avoidance tests. So these are how shy is your mouse or how anxious is your mouse. You can use whatever anthropomorphic terms we want, but the amount of time the mouse spends in the open arms reflects its anxiety. So if they spend more time in the open arms, they're less anxious. And in the bottom, we see the data related to this. So these germ-free mice, in comparison to some normal mice, which are called specific pathogen-free, they spent more times in that open arm. So we took away the bacteria, we took away the anxiety. And then if we take a look at the activity, so just how much time do they run back and forth between the closed arms, so this has 2 open arms and 2 closed arms, it was the same in both mice, but they go out on the open arm more often. So this was a bit of a surprise test. We expect to get a stressed out mouse, but we actually got this less anxious mouse. And then what happened was a group in Sweden showed the same thing, and a group in Ireland showed the same thing. So we have 3 different groups across the world showing exactly the same phenotype in these mice. And this was about 5 or 6 years ago, and it's been reproduced a lot. So, getting that sort of consensus across labs, across different systems is really great for science. But the other thing is all the studies to date in people where they've given probiotics or they're starting to look at the microbiome have made these same links. So we're pretty convinced that the microbiota is influencing anxiety. We've also showed a whole host of different brain-based genes that are changed. And in collaboration with Jason, who's going to speak next, we've actually done imaging on these mice to look at brain structure. And one of the things that this graph just quickly shows you without a lot of detail is on the left-hand side, we see the difference between germ-free and normal mice. If you look at absolute volume, and all the red is bigger. So the normal mouse has a bigger brain than the germ-free mouse. So, it builds a bigger brain. And then if we normalize that data, we start to see some very key regions that show up that are different. And so some of those regions are part of the stress axis. So just to summarize the data from the germ-free mouse, we've shown that the microbiome influences behavior, stress circuitry, stress responsibility, brain structure. And a summary that just came out in a review paper by John Cryan's lab in Ireland shows that it has all of these different effects. It has neural function effects, it has stress axis effects, it has neuroprotection effects. So the microbiome is very important to the health of your brain. And we've also shown that those neurons, that part of your little brain that surrounds your gastrointestinal tract, where there's a neuronal plexus that takes care of motility and other things, it's also linked to the microbiota. But the question, million-dollar question, is what's the evidence that bacteria itself is having anything to do with the behavior? So the way that we've started to look at this is we actually can collect the fecal samples and sequence them using current sequencing tools, and we can show that there's these different distributions of bacteria, and this is the behavior of the particular mice that we did in this experiment. And the question is, are they associated with this behavior? And if we just take a look at this open arm time, which is one of the outcomes there, you'll see that if we actually map out the individual mice, there's a fair amount of heterogeneity in anxiety-like behavior, even though the mean looks pretty clean. And so if we actually map this onto those different species of bacteria, we actually now have found 3 bacteria that we think are associated with that anxiety-like behavior. And those bacteria, interestingly, are fairly abundant in a mouse bacteria. They have homologs in a human, but 2 of them are actually, the first 2 there, are considered anti-inflammatory or protective bacteria. And the third one, interestingly enough, is one of these bacteria that's associated with cancers and is thought to increase inflammation in your body. But when we look at the way that they're related to behavior, we actually see the relationships between these bacteria going in the same direction of increased inflammation being linked to increased anxiety. And that's sort of summarized here in a hypothesis we put forward a few years ago, that one of the ways that the bacteria might be influencing your brain is by driving the inflammatory state of your body, which then, of course, can communicate with the brain. And so that's what this picture here shows. And I'll just run through, this is just to provide you with some of the information of how this might happen quickly. So we have neural connections of the autonomic nervous system and the enteric nervous system that could do it. We have blood-borne signals, which are those pituitary hormones I talked about that are released into the blood, but also your gut and your adipose tissue can release gut hormones into the blood. They can talk to the brain. We have cytokines and immune mediators when we have an infection or even when we're healthy that can be released by a variety of cells into the blood that can talk between. We have immune cells themselves, which my lab's very interested in, macrophages, monocytes, T lymphocytes that go back and forth between the gut and the brain in communication. There's all sorts of metabolites. This is important. I think there's a question from one of the parents about diet. Certainly, diet influences microbiota, and that can influence what's the profile of metabolites that are being released into your bloodstream. And some of these are very important in how we signal the brain and how we interpret those signals in the brain. And then finally, your bacteria in your gut can, in fact, make neuroactive chemicals. It makes serotonin. They make trophic factors. So the question is whether any of this production of chemicals in the lumen of your gut is influencing brain function. We don't have the answer yet. And I think I was asked to say if I had unlimited funds to find the biological answers, what I would in fact do would be in mice and in kids across the lifespan, because I think the microbiome is one of those things that would tell us why some of this stuff is temporally driven. Why do these, why are they good here and then they get to adolescence, they switch, would be actually to map this out in full detail in a large cohort. And that means we know how to do it. We know what to look for in urine. We know what to look for in fecal samples. We know what to look for in the blood. We know how to image the brain, and we know how to measure the behavior. So if we could have that in a cohort that went from birth through 30 years of age and then potentially later on, that would be what would would change the field. This just tells you we have a lot of work thinking about how these mechanisms of gene environment interaction take place. And I'll just end by thanking my group that's done a lot of this work and my collaborators as well. Yeah. So I realized that, I listened to what I said last year, and I didn't realize I was being videotaped and being on the web forever. I just attended a probiotic meeting last week. So there's a lot of probiotics available, and it's actually a field that's being advanced exceptionally well. There's a lot of biology understanding. So there's several probiotic companies who are actually testing what is the benefit of individual strains down to different, even if they're all lactobacillus rhamnosus number 1, 2, 3, 4. So probiotics on the market share some commonality. The lactobacillus, which is a very common commercially available bacteria, was the first one that Eli Metchnikoff, the Nobel laureate for blood-brain barrier, actually, and the people who developed the first yogurt used to make the first yogurt. So for 100 years, it's had prolongation of life. So it's a good one. Bifidobacteria is an interesting bacteria. It doesn't appear in a vaginally born breastfed baby till about 9 or 10 months of age, but it's one of the ones that you'd find in a normal probiotic. So there's sort of those two families that are the best characterized to have benefit, and clinically they're the ones been demonstrated to have benefit. Interestingly, my list didn't include those. It included 2 novel ones, so hopefully we'll get some more. But the ones on the market, if they're alive, if they're refrigerated, and they're at high levels, you want as many as you can get, they're all pretty good. The liquid ones, the BioK, sometimes is tolerated more. They have a variety of formulas, and it's a very good one as well. It's probably the best quality as far as that. And it's some of these are enterically driven. You have to look to see that it's going to make it through your stomach and land where it needs to act.
But basically dark bottle, refrigerator.
Yeah.
Dark bottle.
Dark bottle and refrigerated.
And refrigerator. Stuff that's sitting on the shelf of your supermarket.
Yeah.
That.
Yeah.
Dark.
Yeah.
Yes. So you light exposure is a problem.
Yeah.
My son has been taking the probiotics for over 7-8 years already. So my question is, would the billion counts, then CFU would matter? Because I'm taking as high as 110 billion counts versus 30, 40, 50.
Yeah, so one of the reasons why I showed that slide of all the different things that can affect your microbiome, right? So probiotics are one of those. And so often people ask me, do I take a probiotic? And I don't. I actually think that my diet and whatever else I do produces this healthy microbiome, right? So, or it works for me. So probiotics are good, as I said, but if that's not where the problem lies, then, it doesn't hurt to take a probiotic, but if it's not where all of those different levels that I showed in that previous slide, whether it be the immune system, the gut bacteria, the cytokines, inflammation, if you're not in a higher inflammatory state, maybe it's not going to have an impact on your own symptoms, right? So that's where the heterogeneity comes in, that maybe it's helping and you don't know that it's helping, but it could also be that it's not targeting some of the deficits because it might be more than one deficit. Yeah. That's what Avdikia does. But yeah.
I mean, that is a very good question, and we don't know how to do it well yet. So all of those tests that some of you do that send fecal samples and get back types of bacteria, I can tell you we have no way to interpret this. I don't know how people think they're interpreting this. There's quite a bit of variability in the normal gut. We find differences between siblings. We find differences across a variety of settings. The clinical risk markers for not having enough of a diverse microbiota for your gut is the picky diets, right? So the more diverse your diet is, the healthier the complement of microbiota in your gut. If you eat one thing, you can be sure that you don't have a healthy complement of microbiota, right? So typically what we would advise parents is for kids who are picky eaters in terms of restriction, so not having variability, and kids who are actually fed with supplements, the kids who take only the bottles of food for a variety of reasons, because there's absolutely zero variability in that kind of diet, we typically suggest that they get supplemented with probiotics.
I just add to that, the other thing is social behavior is one of the domains that microbiota influence as well. So that's part of what this program is targeting, and that's why I put that mechanistic, if you gave me, well, you'd probably have to give me $5 million, that would be where I would dig in to associate not just who's there, but who's it talking to and how can I link it to the behavior. Yeah, at the back.
Yeah. Is this research have anything to do with at all lactose intolerance versus food intolerance?
So it would, because in the long run, some of the risk factors that are associated with food allergies and lactose intolerance are probably being developed in those early years where your microbiome's being laid down. The question is how much of that, so in those people where they have lactose intolerance, is their composition more driven by the environment, or is it more driven by, and I said in a healthy mouse, or in a healthy human, 45% is there because of our DNA we got from our parents, and 55% is because what we ate and did in our early life. But in people with, let's say, lactose intolerance, is there a shift so that more of their microbiome is linked to their diet and less to their DNA, or vice versa? We don't have those answers yet. Well, whether it be, again, there's a mix between gene and environment, and so I'm not sure that we know how much. I don't think we've asked the question yet about that balance in clinical populations, right? So we treat everybody as the same, so if we could actually determine, are you a person who your gut bacteria or your immune system is driven mostly by your DNA and less by what you eat, then maybe we could figure out whether or not, which is, because in some, you could have a food intolerant person who's DNA driven, and you could have a food intolerant person that's diet driven. I can't think that the same therapy would work for both of them.